Good afternoon, everyone, and I welcome una back to our channel today. I hope say all the videos where we dey put out now they understand all these things where we dey talk, and as we dey talk now they gain better understanding to help for classes and different things we una dey do. Today we will discuss about viruses. But before we go into this discussion, I just want to give shout out to one of my oga, Professor McDonald Godaro. In they watch all these videos and in they provide corrections and suggestions for us to improve the quality of the thing we will do, and I appreciate them very well. Thank you very much, sir. Today we're going to discuss about viruses and wetin viruses be. Now viruses dey important because viruses then dey between living things and non-living things. If we look through some of the videos where we don't put out, where we dey discuss, we go see say we don't talk about the most recent method where scientists they use classify organisms, and Robert Whittaker been designed this method, and we dey call am the 5-kingdom method of classification. And this 5-kingdom method, if you notice, you go realize say viruses no dey for that 5-kingdom method because viruses dey problematic. They give scientists problems on the correct way to classify them. You know, they fit classify them as living things or classify them as non-living things. Now, if you see viruses, one of the things to notice now, say viruses dey microscopic in nature, meaning say you need microscope to fit view them. You no go fit see them with your naked eyes except say you use microscope view them. Now the interesting thing be say no be just ordinary microscope you go fit use to view these viruses. E get the kind of microscope wey you fit use to view viruses, and that kind of microscope they call am electron microscope. This electron microscope na very powerful microscope. Many microscopes dey, light microscope dey, hand lens dey sef na microscope, compound light microscope dey. But to view viruses, because dem dey very small in nature, na electron microscope we dey use. And that electron microscope dey powerful because when you dey talk about if you dey talk about microscopy now, e get two major things wey we dey look at for microscopy. The first thing na magnification. That's how big is the object going to be. That thing wey you dey view with the microscope, the image wey the microscope dey produce, e go dey much bigger. So by how many times? That's what we call magnification. The second thing we will talk about for microscopy na resolution. And wetin resolution mean na say if you put two objects beside themselves and you dey view them with microscope, shey you go fit differentiate say these two things na two different objects dem be. If you fit differentiate, then it means say that microscope get high resolution or get correct resolution. So viruses na electron microscope we go use view viruses. Now one important thing about viruses na say na only if dem dey inside living cells they dey active, they do different processes. But if they dey for environment, they dey inactive, they no dey fit do anything, and they dey take on form wey we dey call crystalline form. You know, like your salt na in the form of crystal. So they dey that crystalline form like that if they no dey inside living organisms. Now sometimes viruses we dey refer to them as acellular entities. We no dey call them cells because they no be cells. E get different things wey cells dey do wey viruses no fit do. So in order to just give them a particular name, we dey call them acellular entities. Now make we look at some of the characteristics wey virus get wey dey important. One, dem dey microscopic in nature. Now secondly, dem get wetin we dey call genetic material. We don discuss DNA and RNA before. Now viruses dey get either DNA go dey that virus or RNA go dey that virus. If na DNA dey the virus, we dey call the virus DNA virus, and if na RNA dey that virus, we will call that virus RNA virus. So this one dey very important. Viruses then get genetic material, either DNA or RNA. They no fit get the two for inside the same virus. Now apart from that, they can't get one structure wey we dey call capsid. That capsid, another name wey they call am na protein coat. Capsid or protein coat, e cover the whole of the virus, and na the major structure wey virus get be that. E get genetic material and the protein coat wey be capsid wey cover am. Now this protein coat dey get many different shapes. E dey give viruses their shapes. E fit get helical shape. You know, helical shape na the shape of DNA. This spiral shape wey we don discuss before, that one na the helical shape. Some capsids dey get this helical shape. Some other capsids dey get shape wey we dey call icosahedral shape. Now this shape dey interesting. E dey a bit complex, but na shape I go show you the picture. And na shape wey be say the shape of the virus go get like 20 different faces and those faces na equilateral triangles. Na triangles dey those different faces, and na 20 faces like that. Then some viruses dey get wetin they call complex shape, so you no fit fully describe wetin their shape be, but na the protein coat, the capsid, na im dey give viruses this their shape. Now another thing to understand for this protein coat na say the protein coat, the capsid, e dey made of by very little proteins wey gather together, you know, to make the capsid. And those smaller proteins, or wetin we dey call subunit proteins, their name na capsomeres. Capsomeres. This one dey very important to note. Now viruses, they no dey undergo reproduction like other cells. They no dey do binary fission. That one no dey occur. Viruses, they no dey respire, they no dey feed, they no dey excrete, they no dey do all those things. But viruses, some of them bin get wetin we dey call envelope. Now if you divide viruses into two, you get viruses wey get envelope, we dey call them enveloped viruses, and we get non-enveloped viruses. Now wetin this envelope be? Envelope na lipid layer. If you remember, lipid na our fatty acids. Lipid layer wey surround the capsid, wey dey all around the body of the capsid, for outside the capsid. Remember, say the capsid na the protein coat. So any virus wey get this lipid layer all around the capsid for outside, we dey call that virus an enveloped virus. Virus wey no get that lipid layer, we will say na non-enveloped virus. Now that lipid dey important too. E get medical significance or medical importance. Any virus wey get this lipid around this envelope, e go dey more virulent, more stronger as a virus than the virus wey no get this lipid layer, this envelope around it. Now e dey also important for us to realize say we dey classify virus in many ways. One of the ways na to classify viruses as either animal viruses or plant viruses. Wetin this one mean? We realize say viruses, if they colonize animals, those viruses wey dey colonize animals, you no go dey find them for plants. And the ones wey you dey find for plants, hardly you go see them for animals. So that one come make it dey easy for scientists to fit, you know, classify them, say okay, these ones na animal viruses, these ones na plant viruses. Examples of your animal viruses, simple example, na your rabies virus. Many of us know about rabies disease wey dey affect dogs. Now also, as animal virus take dey, na so plant viruses dey. So those viruses na for inside only plants we go fit see them. A very simple example na cassava mosaic disease or cassava mosaic virus wey dey cause cassava mosaic disease. So all these kind of things been tell us say okay, e get easy way to classify viruses. We fit classify them on the basis of animal viruses and plant viruses. Another way to classify viruses na on the basis of the genetic material wey dey carry. So viruses wey dey carry DNA, we will call them DNA viruses. The ones wey dey carry RNA, we will call them RNA viruses. Simple example na the HIV virus wey dey very popular. We know say na RNA virus. So this one dey very important. Now another thing to realize na say, I don dey talk am before, say viruses dey only functional if dem dey inside living cell. If a virus find itself inside a living cell, e go dey carry out different activities, e go dey replicate, dey do many things. But if e dey for the environment, if e dey for soil, if e dey for water, if e just dey on top a table, that virus go dey in the form of a crystalline form, and I no go dey do any activity for that form. Now e get wetin we dey call viral life cycle. That's, e get cycle wey viruses dey follow from one particular stage to another stage, and they dey follow that cycle for all the activities wey they dey involve themselves in. That cycle na attachment, entry, replication, assembly, and release. So wetin dey happen for all these places now? Viruses at the end they get wetin they call spikes or glycoproteins. So some structures dey the body of the virus wey dey point out, and they use those structures to attach themselves to living cells. So if they want enter cell now, if they want enter a particular cell, maybe na human cell, they will go attach, that's the viruses, they will go use those their spikes, those their glycoproteins to attach themselves to those living cells. Now that one na the attachment place. From attachment, entry come dey. The next thing na entry, and that entry na say the virus go come inject in viral genetic material, whether na DNA. Remember I tell you say na either DNA or RNA. So whether na DNA or na RNA, the virus go inject that material into the living cell wey e wan colonize. That one na entry. After that one, replication dey. You see, as this viral DNA or RNA dey inside that living cell, you go notice say that living cell get im own genetic materials. The living cell go get im own RNA, e go get im own DNA. Now, this viral DNA or RNA wey enter that living cell go come begin dey use up all the genetic materials wey the living cell get. E go dey use those materials to dey form im own viral DNA and viral RNA. So that way, e go dey use up all the resources wey that living cell get, dey form many different copies of its own, that's the virus own DNA and its own RNA. At the end of the day, e go use up all the genetic material wey that living cell get. Now, apart from the genetic material, the protein too wey dey inside that cell, the viral DNA go dey use up that protein, dey use am dey form new capsids. Dey use am form capsids around itself. So that one too na replication. Now, after replication, we go come reach assembly. E go assemble that capsid. So the virus don dey now, new viruses don dey inside that living cell. After that, the process of release go come, wey be say all those viruses go burst forth from that living cell, and they don destroy that living cell as e dey. They go burst forth and begin to continue into the environment. Now, viruses, they get medical importance and agricultural importance because viruses, they cause a lot of diseases. This COVID-19 now we just pass, no say na virus. HIV na virus, rabies na virus. So many things dey, many diseases dey wey viruses dey cause. That one make virus dey get serious medical importance and significance. They also get, that's the viruses also get agricultural importance because they cause many plant diseases, many animal diseases wey dey lower the quality of agricultural produce. So all these things dey important now why we dey study viruses. Thank you very much for listening to this class and have a wonderful day.